Okay, hello everyone. We are about to start the next section. Is in this case is module 2.2, implementing viral infection model in CC3D, creating virus field diffusion and diffusion solvers. Again, I've posted the link in the chat for people who want to have access to the files that we will be working with in this simulation, in this module. Particularly, we will start off from the module 2.1 version 1, which is where we end at in the previous module, and we're gonna build off from that one to include virus field and putting our diffusion solvers into the simulation. So again, Giuliano is uploading zip versions of the folders. They should be up here in a second. So what I'll do is I'll just give a couple of minutes so people can get them and upload them. The first part of this section is gonna be rather explanatory in the sense that I'm gonna talk a little bit about what diffusion is. So in that time, while people listen in the background, please make sure that you can download the versions and upload them into NanoHub. So in the previous modules, we have already cellularized the transitions in the epithelial cells from uninfected to infected, from infected to virus releasing, and from virus releasing to dead. In this module, now we want to add the virus field and the cytokine fields. And then afterwards, we want to have our transition from uninfected to infected cells be determined by the local concentration of virus rather than the virus in the ODE model. And I will talk a little bit about that later, just as a refresher of what we did when we implemented this transition in module 1.6. In module 2.1, and we will explain what it means to read of the virus from the field as opposed to the ODE model. So the idea is, the basic idea is that when we implement the virus infection, we implemented it in a way that the virus is provided by the ODE model. And what we get as a result of that is that the infection probability is pretty uniformly distributed in our cellular domain. So there is really no special, the virus doesn't have any special property. It's what we also call a well-mixed system, in the sense that the virus is well-mixed, is equally distributed in our simulation domain, which means that the probability of infection is also uniformly distributed in our simulation domain. When we localize the virus, though, we go from that well-mixed assumption to a simulation where the transition between uninfected to infected is going to be locally determined by the amount of virus that we have. And what we get as a result of that is this plaque-like infections that actually infection spots that actually resemble what is observed in experiments when a when cells are infected in, let's say, a petri dish. So this is important because this transition is going to allow us to actually represent what happens in the experiments when cells are infected. So to start with, to a little bit of the background for this is that we need to understand that in physical system, things move because of various different reasons. They can move because of advection. They can move, meaning that the things that have to move along. They can they can move because active transport. For example, if you have cells that are ciliated, that they have cilia on their cell surface, those cilia can actually move stuff, let's say, from one part of the tissue to the other one. But we also have transport by diffusion. And in diffusion, what happens is, as you can see in this image, is because of the random movement of molecules. When you start with molecules are on average going to move from areas of high concentration to areas of low concentration, and the result and the final what diffusion is going to produce at the end is that it's going to erase any differences in the concentration in your domain. So in this particular case, at the end, you can see that these dots are equally distributed in the domain because they have started moving due to diffusion. Diffusion is very important in biology. It's one of the main transport mechanisms in cellular systems, especially over short distances. And the reason it happens, it's important over short distances is because the distance that a diffusive particle moves is proportional to the square root of time. So for example, if it takes 1 second for a molecule to move 1 micrometer, then it takes 4 seconds to move 2 micrometers, and it takes 100 seconds to move 10 micrometers. So as cells diffuse for larger distances, it takes much more time. So that is why it's very important in shorter distances than in wide range, in larger distances. When cells encounter processes, when cellular processes require movement over large distances, they would usually involve active transport processes. But over short distances, cells relied on diffusion because it's cheap. The cell doesn't have to add any energy, doesn't have to use any of its energy to transport cells from one point to the other, because cell molecules will just move again from areas of high concentration to areas of low concentration just by wandering around. So it's a cheap transport mechanisms for cell, and cell take advantage of that to have access to different molecules. In our particular case, right, we're interested in small molecules like viruses and cytokines, but we are not going to represent virus and cytokines as individual molecules. what we're gonna do is that we're gonna assume that there is enough of those molecules that we can treat them as a density, as molecules per area, rather than having to model individual particles at a time moving, let's say, in the extracellular environment. We're going to assume that they behave as a field, in the sense that we can describe them as a density, which means that we can describe this in terms of number of molecules uh, per volume. And that this means that we're going to treat them as a continuous quantity, whereas in reality, there's viruses are discrete quantities, right? There's one virus, two viruses, 1,000 viruses. But when there's enough viruses, then we can say, okay, we're not concerned about one specific virus. We can treat them as a continuous quantity. Same with the cytokine molecule. In CompuCell, we represent these fields as scalar quantities associated with each voxel in our simulation. And we explained yesterday that the voxel is the minimal space unit in CompuCell. It describes this minimal space in CompuCell. So what we do is that we associate with each voxel a number that says what describes the density of the field, let it be the virus or the cytokine, of the density of the field in that particular voxel. There's also a possibility, this is in the case when we treat them as scalars, but there's also the possibility of treating them as vector field objects. That is quite limited. We're gonna for this particular model, we're gonna be using the scalar field objects again, which means that at each voxel we're gonna have a number that describes the density of cytokine and the density of virus at that particular location. A good question. So whether does this account, the question is whether this accounts for varying vital load. So the quick answer is that we're gonna assume that we have enough virus that we can actually make the approximation that we are doing here. If there is very, very small amount of viruses, then we might not need to take a different approach in terms of how we represent viruses. But we're gonna assume that there is enough of them that we can treat them as a continuous quantity. Good question. So yeah, now I explained that the way we're gonna treat these fields, the first thing I explained is that we're gonna treat our viruses and our cytokines as fields. And I explained that the way we do that in CompuCell is that we associate with each voxel a number that describes the density at that particular voxel of the chemical field that I'm talking about. And now the question is, how do we determine how that density or that concentration changes at a particular voxel? And the way we do that is by using the diffusion equation. And we're not gonna get deep into the weeds of the math here, but the idea is that the diffusion equation is gonna describe the change in concentration of the field at a particular location, at a particular voxel. And it has 4 components. It has the component associated with the diffusion itself, that is with the gradient at the field, at that particular field, at that particular location. And by gradient, we mean how the concentration is changing with respect to the location. It's going to have a decay constant. So I'm sorry, that how the concentration gradient, how affects the diffusion or changes the concentration is going to depend on this term, the diffusion coefficient. And the diffusion coefficient indicates how diffusible a particle is. So if the particle is highly diffusible, then it means that the concentration at a particular location is going to change rapidly. If the particle is non-diffusive, then the concentration is in the field is not going to change because of differences in gradient. So this term is basically accounting for the fact that molecules move from areas of high concentration to low concentration due to just random walks or just wandering around. But as we see, it's dependent on the diffusivity of the field, of the chemical. We have a decay constant that is just how stable the molecule is at a particular location. So if the molecule is highly stable, then the decay constant is going to be small. If the molecule is highly unstable, then it's going to have a large decay constant, which means that the concentration inside a field is inside a particular location is going to go down quickly. There is also an uptake that we're actually not going to go into details here. I'll explain why later. But the uptake, it means how much at a particular, in the particular context of CompuCell, means how much a cell is consuming, how much of that molecule or that field the cell is consuming at that particular location. Remember from our discussion yesterday that cells are composed of many of these voxels. And what the uptake will do is would account for cells taking out from that field because cells are consuming chemicals. And conversely, we have a term that accounts for the secretion of chemicals. And because cells both uptake and release chemicals to their environment. So we do have to account for how the concentration of the chemical changes because of this uptake and release of the cells. And this release is going to be important because this is how we're going to add virus later on, because cells are going to release virus to the environment. Okay. CompuCell has its own ways of basically calculating how these concentrations are changing at each location, and CompuCell does it by using diffusion solvers. There is 3 diffusion solvers that are constantly supported. There's more of them available, but the ones that are mostly supported are the steady state diffusion solver, and these are used only in cases where that diffusion coefficient that I explained later is very high. That in cases where the diffusive molecule is, or the molecule is very highly diffusive. The reason we use that particular one is because otherwise computations of that diffusion equation can become very expensive computationally. Diffusion solver FE, which is the one that we mostly use, is used for transient diffusion, and it supports it supports specifying diffusion, and we'll see it later, specifying diffusion coefficients at different locations of the different diffusion coefficients at different locations of the simulation domain. And same for coefficients, decay constants. And the last one is the reaction diffusion solver FE. That one, it's just like the diffusion solver, but it adds reaction terms such that we can couple one field with another. So for example, let's to include antibodies in our model. We would include these antibodies as another field. So we would have our virus field, our cytokine field, and our antibody field. And we know that in biology, in the biological system, antibodies bind to viruses, and that's how they disable them. So we can actually have a reaction happening in CompuSol between the antibody field. And again, this is not, we're not gonna walk through this, but it's just an example, and the virus field. And this is something that we could, if people are interested, we could also develop, talk a little bit more during the workshop, during the hackathon. This is a question that is more for other people in the lab than for me. I would pass it along. There is a question as to whether the ECM would be appropriate to represent as a field. I'm not gonna go into details, but other people in the lab might be able to answer it in the chat question, in the chat section. So that was a quick and dirty explanation, quick and dirty walkthrough of diffusion in CompuSol. Again, the main idea is that we're gonna represent our virus and our cytokines as chemical fields, and then we're gonna decide we're gonna decide whether cells get infected based on the local concentration of the virus field. Okay, so the first thing we're gonna do is that we're gonna load our project. We're gonna launch CompuSol in NanoHub, and we're gonna launch to edit by hitting File, Start to Edit. Once we're in Edit, we're gonna open a project, CC3D project, open CC3D project. We have to go to a home directory in NanoHub. And in this case, as I mentioned, we're gonna be working from module _ 2.1 version 1. We're gonna build our simulation from that one up. So I go inside the folder and I always open the .cc3d project to be able to launch the project. And again, that will load a project into CC3D. We can open all of our documents by double clicking on the .cc3d file in the CC3D simulation tab here on the left. And this is basically the simulation that we ran at the end of the previous module. It has all of these plots, and we explained that we are tracking the number of uninfected cells, the number of early infected, or the number of virus releasing, and the number of dead cells. And we explained that at the moment there is a difference arising from the fact that we are not accounting for the innate immune response, which is fine. We will add that later on, but we know we can explain where the difference is coming from. So we're gonna walk through adding more complexity to the model, and then we'll come back to this later on. Okay, so the first idea is that chemical field specifications are done in CompuSol, are done in the XML file. we can access this XML file here. And the way we are going to include we need to be able to include our current XML file does not has any chemical field specified on it. And we saw when we talked about this in version 1.6, we saw that Wizard has the capabilities of allows users to predefine chemical fields in Wizard. But at the time, we didn't needed them because we wanted to focus on the cell transitions first. And now we're gonna start adding the chemical fields. So we can, if we did not add them in Wizard, we still do have the option of adding them later on. The way we will do that is we will go to CC3D ML, which contains the templates for the XML file. We're gonna go to Steppables, and Steppables are gonna show blob initializer. We did talk about that one a little bit, but it's also gonna show, which allow us to bob initializer and uniform initializer allow us to initialize cells from the XML, but we already initialize cells in Python, so we don't need them here. But we do have available multiple diffusion solvers. Some of them I did explain already: the steady state diffusion solver, the diffusion solver FE, and in this particular case, we are gonna use the diffusion solver FE. So I click on that one. And the default code that gets imported into XML is actually quite elaborate. It has a lot of options for users. Some of them we will not need. I will walk you through how to do model specifications, chemical field specifications here. But it's important to know that it gives you the default code gives you the option of initializing two fields. What is called field name 2, field name 1 here, and field name 2. What we're gonna do is that we're gonna start working with the first chemical field, which is gonna be the virus, and then as an exercise, you guys will follow the same workflow that I'm gonna be explaining now to implement the cytokine on your own. Okay. So that's important. If you guys can follow through what I'm gonna be doing here, that's gonna come handy later when you implement the cytokine by yourself. Okay. So the first part of this template code specifies what is the diffusion solver that I am using. And then it allows me to name the chemical field that I want to include. So in this case, we want to include the virus as a chemical field. So we're going to change that. And it's also specified down here in the category field name. We're going to change that one as well to virus. Okay. Next, we can define the global diffusion and decay constants. And as I explained, those correspond to these parameters in the diffusion equation that is going to be used to calculate how the concentration changes in our whole domain. For now, what we will decide the values of the diffusion constant and the decay constant in Python. So at this point, it doesn't really matter then what value we put in here. What we do want to add for now is an ID that is gonna allow us later to manipulate these values from the Python script. So we're gonna add an ID called virus DC, an ID called virus decay. And again, as I mentioned, what numbers are here at the moment don't matter much because we will change those later on from Python. Besides this global diffusion constant and global decay constant, the diffusion solver also, as I explained later, it gives us the options of defining different diffusion coefficient and decay constant by cell type. And what does this mean? So from a biological point of view, this will mean that a particular molecule is more or less diffusive when it's inside a cell. So let's say the cell is much more crowded, the molecule will be less diffusible. Also, let's say it binds into something inside the cell before being transported outside, that might also change its diffusivity. It's now, it can be either. It can be single or double quote, as long as it's a string. The decay coefficient inside a cell would represent something like the molecule being more or less stable inside a cell. So if the cell is consuming a molecule or using it up for something, then, or degrading it, then the molecule will have a different decay coefficient inside a cell than it has outside of the cell. At the moment, we're gonna assume that the virus is equally diffusive across our simulation domain, and it's equally stable across our simulation domain. So we don't need to specify different diffusion coefficients or decay constants by cell type. So we're gonna just delete that part. But I wanted to explain what would be cases in which you might want to use that. And again, our assumption that the virus is equally stable across the simulation domain, it could be wrong, but it's important to know that that is the assumption that we are incorporating into the model. The XML, the XML specification also of the chemical field also allow us to predefine an initial concentration of the field. Let's say that at the beginning of our simulation, we want to have some field in some amount of the field of the chemical in our simulation domain. We can specify that here. It can be either a function of the locations in the simulation domain. So for example, it could be higher when x is 0 and then smaller when x is, let's say, 100. Or we could also import a file that has specifies for each location in our simulation domain what is the concentration of the field at that location. And in that case, the file will look like something like this, where we specified how much of the virus or the chemical field in general, but in this case, the virus is at each particular location of our simulation domain. Okay, so we're not gonna specify any initial concentrations of the field. So we can also delete this part. It's actually commented out. I'm just gonna leave it because it's already commented out. Secretion data. So as I mentioned, cells can secrete and uptake from these chemical fields. And this here we will specify the rate at which cells are taking up from the field if those rates are if those rates are constant. There is also other options to how to specify secretion. In this case, secretion can be uniform, or secretion can also be on contact, which means that cells will secrete or uptake only when they are in contact with only when increasing when they're in contact with other cells. And James just made a very important comment about how to include comments in XML. If I wanted, instead of deleting what we've been doing is deleting, because probably it's one of the safest options. It also clears up a little bit of the redundancy that we need. But if we wanted to comment it out, we can always come here again to edit, block comment, and that will comment out the portions or sections of the code that I do not need. But you have to be careful, because if you add a comment on top of a comment, for example, this will cause an error and it will cause Compusel to crash. So we need to be able, if you're gonna comment out, you need to make sure that you're not double commenting stuff. Thank you, James, for the reminder. So, yep, we're not gonna specify secretion, although our cells are in fact gonna secrete virus. We're gonna specify that secretion in our Python Steppables script, so we don't need the secretion data either. Just gonna delete it. Just so I'm just you can comment out, but I'm deleting just to make the code more compact. Next, we need to specify boundary conditions. And by boundary conditions, we means what happens to the chemical field at the edge of the simulation. And there is at least 3 boundary conditions that we can specify. We can specify constant value, like this one. And in that case, what we mean is that at the edge of the simulation, the value of the field is gonna be always the same. So in this case, we have to, and also we have to specify boundary conditions in every one of our dimensions, right? So what happens when the field reaches the edge of the X dimension? What happens to the field at the edge of the Y dimension? What happens in the Z dimension? So, and then we have the same, we can vary or combine these options depending on what we want to represent. Let's say we have a source that we want to represent at the edge of our domain, let's say because we want to represent a blood vessel that is going parallel to our simulation. We could represent that as a constant value or as a constant derivative, in which case it's either it's always the same value at the edge, or the edge is actually producing or secreting into my simulation. In that case, I would use the constant derivative. Um, Depending on the specific biology, you can choose one over the other. For a particular simulation, or you can have periodic boundary conditions. In the periodic boundary conditions, we are making the assumption that our simulation represents just a portion of a bigger space, but that every other space in that bigger space is just a reflection of what's happening in my simulation domain. So if one molecule, let's say it's going out in, let's say, this direction, then the same molecule or another molecule of the same type will come into my domain on this direction. So it's reflected. So if this molecule goes up this way in this dimension, then this one will also come up. So basically what goes out in one end comes in at the opposite end of my simulation. What we're gonna do for our simulation is that we wanna, because we are assuming that we're representing a patch of tissue, right? Our patch of tissue is, we can assume that is periodic in the X and Y dimensions, which would represent that we're just looking at a small fraction of a bigger epithelial sheet. But in the on the C dimension, there are differences because we are representing, one is the extracellular environment, whereas the other is either more of the cell, or let's say another part of the structure of the tissue, like the ECM or stromal tissue underneath my epithelial sheet. So based on those assumptions, what we will do is that we will define periodic boundary conditions along X and Y dimensions. So we have to delete the options included with the template code and uncomment the periodic boundary. In both the X and Y dimensions. And on the C dimension, what we want is constant derivative to be 0 in both cases. What that means is it's also known as no flux because nothing crosses the boundary. So basically, everything that goes against the boundary bounces off and comes back to the simulation. So we don't want any flux going up and down the simulation. So we're gonna set it to a constant derivative of 0. So basically, in the C dimension, everything is enclosed, but in the X and Y dimension, whatever goes out comes out the other way. And that finishes our field specification in XML. Just a quick recap. We have the name of the field, which is the virus field. We have specify IDs so we can manipulate diffusion constant and decay constant from CompuCell, from the Steppable, Python Steppable script. And we have specified what happens on the boundary of the boundary with this field. And we said that it's periodic across X and Y, but it's no flux in the C dimension. And I'm gonna stop there for 2 or 3 minutes to give people chance to manipulate the codes as well. And if, in the meantime, if there's any questions, feel free to drop them in the group chat. We're just gonna wait 2 or 3 minutes. So in the C dimension, the code already has, the template code already had the constant derivative as an option. So what I did was I changed the values, I think the original values that came in with the template, and so I set them both to 0. Okay, so seems like most people have been able to work on the exercise or follow through. So now that we have specified the field, actually, if we come back to CompuCell and save, we saved, and we come back to CompuCell and relaunch the same simulation that we're working on, file, open simulation, and a hub directory. We've added the field, but we haven't done anything with it. There's nothing happening with it, but we can see our graphic window if we choose the pull down menu. If we choose the pull down menu, we can see that now we have the virus as a chemical field that we could display, but we wouldn't see anything because we haven't initialized any virus, or cells are not at the moment secreting virus. We just have it as a placeholder at the moment. Okay, so we will continue working with this. So as I said, as I mentioned, the diffusion parameters, both the diffusion coefficient and the decay constant, can be specified in Python, in the Python script. So now we can go back to our Python script. And what we're gonna do here is that we're gonna define our virus diffusion constant. In principle, this is a parameter that can be experimentally measured. And we tend to prefer adding experimentally measured parameters as global parameters in our simulation. So that's why we are initializing it here. This number is pretty much arbitrary. We just shows a particular number. But again, this would be an experimental measurable parameter, and then we can we could estimate it from experiments. But we need to define it here as a global parameter. Now, as I mentioned before, what we want is to be able to pass this number to my XML file. So I could, in principle, add it here as well in the XML file where I specify the virus diffusion constant. But again, since this is a experimental parameter and also a parameter that we want to play with dynamically, we can play with dynamically, we want to specify it as in the Steppable script. But now we have to be able to tell XML, this is the number of the global virus diffusion coefficient. To do that, I can use a function called get_XML_element, pass the ID that I created. That's why it was important to include that ID. That's CData. That function is not currently available in any of the scripts of the template scripts, but it can be accessed in the documentation if you look at how to pass information from the Steppable script to the XML file, because that's what we are doing here. We want to pass information from the Steppable file to the XML. So how do we do that? We go to our start function, and we, after loading the anti-money model. So this is we talked about yesterday in 1.6 how to load the anti-money model. We want to pass the data that the variables that we have defined here to the XML. This is going to be called virus decay, diffusion, and decay. And as I mentioned, we can use this function, self.getXML element, specify the ID that we specified in the XML file, which was virusdc, CData, and then the variable that we store the diffusion coefficient value as a global parameter. That is gonna, this line is gonna tell XML, okay, this is the value that I wanna use for my global diffusion coefficient. The next parameter that I need is the decay constant. The decay constant is given by the ODE model, because the ODE model, when we go back to the ODE model, has it has a term that corresponds to that clearance or cell death of or decay of the virus. So what we're gonna do is that in this particular case, the one that corresponds to it is the parameter C. So we wanna tell XML, use the parameter C from the ODE model, from the anti-money model, to determine what the decay constant, or use this as the decay constant for my virus field. So we can use the same syntax, but in this case, the ID that we use was virus_decay. And as we discussed yesterday, the way we access values from the anti-money model is by using this syntax, self.svml, the name of the model, the way I loaded it. And then we can reference values by using the either variable or parameter name in the anti-money model. And this particular one needs rescaling. So we have specified now from the Python Steppable, the Python script, both the diffusion coefficient and the decay rate of the virus. Gonna give people a couple of minutes to do that to be able to finish it. And let me see if there's any questions in the chat. Okay, no questions, good. Okay, just giving people just a couple of minutes to catch up. So going back to our diffusion equation, we have already determined the diffusion coefficient and we have determined the decay constant, but now we have to define the secretion terms, right? Because what happens, cells have the capability of both taking up chemicals from their from the extracellular environment, and they also secrete chemicals to the extracellular environment. And they do that by different mechanisms, either by passive transporter or active uptake and secretion. Those kind of things, we are agnostics in terms of what is the specific cellular mechanism that is being used to uptake and secrete, but we can represent the secretion and absorption of chemicals by the cells. So, and that is going to account in our diffusion equation for this secretion term. So in our model, right, the cells that are releasing virus is the virus releasing cells. Those are the cells that actually secrete virus to the extracellular environment. So the first thing we need to do is to say, okay, I want my virus releasing cells to release a certain amount of virus per unit time. Iterate through my virus releasing cells. And I'll do that in the step function underneath the integration of the ODE models. Okay, the first thing I need to do is to iterate through my list of virus releasing cells, and then once I'm iterating, then each cell is gonna secrete a certain amount of virus. Just as a refresher from yesterday, a reminder from yesterday, we can iterate cells by going to Python, CC3D Python visit, and then we can iterate through all cells or cells of a given type. We're gonna iterate through cells of a given type, and the type is gonna be virus releasing. Again, reminder, cell types are always specified as in all caps in Compusome. But basically, I'm iterating. Now I have to do, now here, my cells are gonna secrete inside the loop. So to have individual cells secrete a chemical field, we need to add a secretion function. And conveniently, the secretion function is something we can access in our templates, in our code snippets. If we go to CC3D Python, there is an option for secretion and uptake. So although we're in this module, we're focusing on secretion, we could actually uptake from the environment as well. And we're gonna go ahead and choose secrete inside cell for now, because we will discuss a little bit later how we will need to modify it. So the first thing this template code reminds us is to make sure that the secretion plugin is loaded. And the way CC3D plugins are functionalities that extend model capabilities. So extends the behavior of objects and simulation objects, including cells, allowing users to specify more complex behaviors. The way we load plugins is that we do it in the XML file. So to meet this requirement, we need to go to the XML, and we can see that we have already loaded one plugin, which was the diffusion solver. We're gonna need another plugin. In this case, it's gonna be the secretion plugin. So I'm gonna just put it before the diffusion solver. The template to include the secretion plugin can also be accessed in our code snippets, in our templates. If we go to CC3D because now we're back into our XML file. If we go to plugins, we see that there is a lot of plugins or behaviors that we can add to our cells and additional capabilities that we can add to CompuCell. The first one that the one that we care about here is a secretion plugin. So again, I go to my XML file. Before the diffusion plugin, I'm sorry, it has to be after, because this is the center of mass plugin. It might or might not be loaded in your simulation depending on what code you're using. But we're gonna add another plugin that is gonna be the secretion plugin. CC3D plugins secretion. The default template includes also basic information of if we want to specify some secretion in the XML, but we are doing that in Python, so we don't need any of the template information that comes with the default code. So we're just gonna get rid of it and make sure that you get rid of everything in that category from field to field. I'm just gonna go ahead and delete everything from here. So basically I have the beginning of the category, and this is in XML how you end the category. So I have an empty category, which is gonna be loading my plugin, my secretion plugin. Okay. So once I've loaded my secretion plugin, let me Okay. Let's go back to the steppable, and now we can delete this because we have loaded the secretion. I'm gonna give people a couple of minutes to catch up and questions. So just as a reminder, we use the CC3D Python options, went to secretion, secrete inside cell, and the template code reminded us that we needed to load the secretion plugin. So we went to the XML and we in the XML, we shows from the template codes at CC3D ML, we shows the secretion plugin. Okay. There's still people working, significant amount of people working on it, so I'm just gonna give people a couple of minutes to finish it. I'm gonna grab some water to give people finish to it, to give people time to catch up, and I'll be back in 2 or 3 minutes. Okay, we're gonna continue now. So if we go to our steppable files, we now have a secretor loaded. That is our secretor functions, secretion function. And we have the secretor. It's gonna make reference to the chemical field that we have, that we want to refer we want ourselves to refer to. In general, this object, the secretor object, allows us to do multiple things related to the chemical field. So it's gonna be a reference to the chemical field in our simulation. And since it's gonna be it's gonna be a reference to the chemical field in the Python script. Since it's gonna be a reference to the chemical field in Python, we need to modify the template code to make it specific to our simulation. And the way we're gonna do this is we're gonna modify the variable we're gonna modify the variable, and we're gonna modify the variable the object is being attributed to, and we're gonna reference the field as we define it in the XML, which was virus, was with a capital V. It's important, it has to be exactly the same way it was defined in the XML. So now we have our secretor virus object, virus secretor. Our virus secretor object that is a reference to the field, the virus field. We do not need the template code says that you might reuse the same secretor for multiple cells, and since that is the case, right? Because we want all of our cells to secrete the virus field, we want to define it outside of our loop. So it's gonna be before I iterate through my virus releasing cells. So what I did was I just adapt, I'm sorry, let me scroll. I just adapted the secretor so it corresponds, it references the virus field that I created in the simulation, in the XML. Okay. Now, if I come back to CompuCell and I make sure that my simulation is saved, it's not, and I go back to CompuCell and I relaunch my simulation. One second. I have a syntax error. The syntax errors might arise from where I am defining stuff. So the problem is that this variable, the scale by time variable, I actually calculate later. So the only syntax error that I had in the code is that I need to specify the vital diffusions parameter after I calculate my scaling factors. So I'm just gonna take this and move it after the scaling factors. I can save my simulation, go back, play. So let's pause for a second. So now, by default, the simulation is opening just one graphic window with the cell field at level 1. The first thing I want to do is to go to level 0 so I can actually see my epithelial cells. And I want to open another graphic window so I can actually see the virus field. The way I do that is I click in this icon to open a second graphic window. And then in the pull down menu, where it says cell field, I can now choose the virus. And it's gonna show in color 0 because there is no virus at the beginning of the simulation. And as I play, and my cells get infected, what I should expect is that they start releasing virus to the extracellular environment. So I have a cell that is infected, but it's not releasing yet, and there is another error because I changed the secretor reference when I was grabbing the secretor, but then I did not change it in the actual secretion function, which is secrete inside cell. So it's gonna be virus secretor. I can relaunch the simulation. Again, I have one graphic window. I open the second one. Every time I open a new graphic window, the simulation is gonna stop. So I just gonna hit play. It's gonna pause. So I just hit play, show the virus. And now we can see the virus is actually being released by that cell as it gets infected, okay? I'm gonna give time so people can see their virus in their simulation as well. In the meantime, the first thing we want to do is we want to be able to control the display options of the virus. The way we do that is we come to configuration in player, and the configurations in player allow me to define the colors of my different cell types. But it also allows me to have display options for my virus, for my fields. At the moment, the default for this particular simulation is, if I choose this pull-down menu, I can choose what field the configurations are gonna be, I'm gonna be modifying. In this case, it's gonna be the virus. And this first part controls how the colors are displayed, how the values of the field are read and mapped onto colors. What the default settings in this particular case, when you fix the maximum range, what it's doing is that it's not reading anything above that number. So everything above that number is gonna be mapped on to red, which is the maximum, which indicates the higher areas of concentration. If we wanna actually see with better resolution, we want the ranges of color to adapt automatically. And the way we do that is by unfixing the ranges. And I hit OK. So at the moment, the cells that produce virus are only the virus releasing cells. So what determines whether a cell is releasing virus or not, the spatial distribution, is where the virus releasing cells are located at. Okay. But as I explained at the beginning of this demo, which cells are infected and which not have no special structure at the moment. So they're gonna be randomly infected in this particular simulation. We're gonna change that later on. We can see that as we change the configurations of the field, now it doesn't look like a big red spot. It's actually rescaling by the areas of the field that have higher concentrations of virus. And that we can see that as more and more cells get transition to the virus releasing cell, we get more and more virus being released into the environment. Okay, let's say we want to, let's say this image is really cool. It does look really cool. So we want to take a picture of that image. The way we do it is we click on this icon that says take screenshot. And that is gonna start generating screenshots of my simulate of the graphical window. I can also tell CompuCell to automatically save my display windows certain amount of simulation steps. And the way I do that is, I'm sorry, I did that too quickly. I come to configurations, and we've explained the cell colors tab, the field tab. But if we come to the output tab, output specifications, we can tell CompuCell to save the graphical windows. The way we do that is we click on Save Image every nth Monte Carlo step. And then here we specify how often, in terms of the simulation steps, we want CompuCell to save the screenshots of my simulation. I can do it as often as I want, and let's say every 10 simulation steps, or as rarely as I want, let's say 1,000 simulation steps. We usually do 10. How often you do it, it's definitely gonna depend on the time resolution of your model. When you click OK, that is going to tell CompuCell to save simulations every 10 Monte Carlo steps, every 10 simulation steps. Okay, so yep. So now it's gonna save images of the plots. These plots are not being saved, but it's saving is images of my graphic windows where my cells and my fields are being displayed. To ensure that I am actually saving the images for these two, I always click on the camera icon just to make sure that the files are being saved. Okay, they are being saved at the default folder. They are saved at is in your home directory. Just now, you have to click on the camera to tell CompuCell which windows you want to save. And once you hit the camera, CompuCell is gonna automatically save the graphic windows that you click the camera on. If we come to our home directory, either your local computer or NanoHub, we're gonna have this CC3D workspace folder. If we go in there, well, it's not saving in this case, but it might be something with NanoHub, but or your computers, you should have local copies of your computer simulation, and then you should also see in inside the folders of the particular simulation that you're running, you will see the images as you save them. Okay, so coming back to the secretion, we can see that in our image that there are areas in the space where the virus is being released, where you have higher concentrations of virus, those that are red, and areas of the space where you have lower concentration of virus. Those are the ones in blue. But we haven't tell at this moment how much they are secreting. It's basically what came in with the as a default in our template, which was 300 units, right, per Monte Carlo steps. What we will do is now is that we need this 300, it comes as a template, but we do have from our model a parameter that specifies how much virus each virus releasing cell is going to release per unit time. And that is specified in the anti-money model by the parameter P. So what we need to do that now is to substitute this P by the amount of virus that I want my cells to actually release, okay? The first thing I do is that I will define a variable called secretion amount that is gonna tell me how much my cell is gonna release per unit time. And that is gonna be a reference, it's gonna be determined by the model parameter P. And again, this one needs to be rescaled. It can be one single double quotation. This needs to be rescaled to our simulation units. Okay. So now we know that this is the amount that I want my cell to release each step. What we're gonna do now is that since we know how much in the cell wants it's gonna release in total, we're gonna change the function that we're using to secrete from secrete inside cell to secrete total count. What this function is doing is that is specifying how much each voxel of the cell is gonna release per unit time. Okay, so this function again is specifying how much our cell is gonna release at each of the voxels that constitute the cell, that gives the cell volume. So since we know that we want to secrete in total, we want to secrete this amount, we want each voxel of the cell to secrete that amount divided by the total of voxels in a cell that I can get from how many voxels a cell has. I can get from CC3D Python, CC3D attributes, cell attributes, volume. And this variable, cell volume, tells me how many voxels the cell has. And we can see that that one only has read access, which means that we cannot manipulate it. We can just read it as a variable. So just to explain this again really quickly, the virus, I'm sorry, it has to be cell the inputs for the function are cell, and then the amount that I want to release. When I wanted to substitute 300, I chose the wrong input. What I want to modify is the second input. The first input is telling this is a cell that is going to secrete in this particular moment. And again, just to explain this, the total amount that the cell is going to release at this time step is going to be P, but the function secrete inside cell total count tells how much the cell is secreting per voxel. So I have to divide P by the total number of voxels that the cell has. So in total, the cell will be secreting P over cell volume times the cell volume of the cell, which is, again, P. It's a little bit elaborate. It doesn't matter. I'm not going to get into the details, but just take in mind that this secrete inside cell total count is gonna release in the this is the amount that cell is gonna release per unit time. Okay, so now we have if we run the simulation again, it's not gonna change anything in particular, but the amount of vitals that is gonna be released to the environment now is the correct amount of vitals, the amount that is determined by the model parameters. Okay, let's just make sure that the syntax is correct. So the last thing I want to do now is I want to be able to compare the amount of virus that I have released to the environment to the amount of virus that is dictate that the ODE says it should be in my system, right? Because the ODE says that there should be certain amount of virus in the system. I now want to make sure that the amount that I have over my whole domain matches the amount that it's in the ODE system. The way I do that, the way I measure the total amount of virus in my domain is using the function secretor that total field integral, right? Secretor here stands for the specific secretor that we want to use. So what I'm gonna do is after secreting, I'm gonna define a variable called net vital total, which means medium vital total. I need my virus secretor. And then again, I can use this function total field integral to calculate the amount of virus that I have over my whole cellular domain. This function is new, so it is still not available in the snippets, but it can be accessed in the documentation. So now this is telling me that this quantity is gonna tell me how much virus I have in my simulation, which is basically going over each voxel and adding up the amount of virus that I have at each voxel. That quantity should match the ODE model if I divide by the number of layers that I have. And the reason we need to do this is because the ODE model describes what it's, the ODE, you can think of the ODE model as describing what's happening just at one layer. So we need to be able to compare to compare the two quantities, we need to measure the amount of field at one layer. And we're doing that by dividing the total quantity by the number of layers that we have. When we have that, we can actually plot it on top of each other, right? And this brings us back to the workflow that we worked with already. It's gonna be a quick reminder here. We go to the area where I define the total diffusive fields. And now I'm gonna add another data series that is gonna be the vital field. I'm gonna delete the. So what I did is I copied the syntax for the vital field from the ODE and pasted it. I'm gonna edit it. So it's gonna be now a different series. It's gonna be the series of the vital field on the ODEs, in the ODE systems, in the cellular system. Those we are plotting with using dots. We're gonna have the same color as the ODE, but we're using dot size instead of line size for for quantities associated with the cellular system. Now I can come back then after measuring the total amount of field in my system, I can actually plot it. I can actually add data points to these data series to plot and compare compare the virus in the ODE system with the virus in the virus in the ODE system with the virus in the cellular model. So to do that, from yesterday, we should remember that because we're plotting these quantities in the log scale, we need to enforce a condition. And the condition is that the variable has to be bigger than 0. Otherwise, the CompuCell is going to crash because you cannot plot 0. 0 is not in the domain of a logarithmic scale, of a logarithmic scale. So we enforce the condition that we're just gonna add data points when the quantity is bigger than 1. And to add data points, once more, CC3D Python, scientific plots, add data point. The handle for the diffusive fields window is met diff data. When the data set that I created was, let's just go back, it was met viral. I always rescale my simulation step so that I can display in real-time units and days to Monte Carlo steps. So it's gonna be plotted in terms of days. And then the quantity that I wanna plot is the total amount of virus in one of the layers of my system, which is this quantity. Okay. This is included with the template, so we don't need it. And let me just make sure that I refer to it the right way. Met vital, yes. Okay, so now I save my simulation, and when I relaunch it in player, now if I look at the window with my total diffusive fields, once my cells start releasing virus, I should start getting some virus in my total diffusive fields. Any difference that we are observing at the moment is because in the ODE models, you can actually have fractions of a cell, and when you have fractions of the cell, those fractions of cells can release virus. Whereas in the cellular version, you have to have at least one infected secreting cell or virus releasing cell to get any virus. But you can see that as one cell got infected, then we started releasing virus, and the virus is now tracking exactly what the ODE says it should be in the simulation domain. So so I have this exercise for you. I'm gonna give people time to catch up, so they can actually display their virus. And then we have an exercise where we are asking you to add the cytokine field following the workflow that we've implemented. I'm just gonna give a couple of minutes to people seems to still be working on it. So I'll just give people a couple of minutes to at least finish up to the point where they have the virus running. And then we can talk about the exercise. I'm gonna take a moment to mention that we have created different versions of the code of this module such that each step adds another um, that each version implements a different part of the module. So if you guys go in, some other people are people are working on it. If you guys finished or if you guys didn't start it, you can always come back to CC3D project, Untitled, open project, and if you guys go to, let me make sure which version is it. If you guys open the version, the one that is the file that says module underscore 2.2 version 2. In that file, we have 3 different folders. In the first folder, virus, that is the simulation that we're just running right now, where we implemented the virus. The next one, we add on top of the virus, we added the cytokine. And the last version of the code is the one where we're gonna talk about what the loop means here. But this is just in case people are experiencing problems that we can follow through the module, and people then can come back and revise their codes against the different versions that we're providing. Okay, so going back to the simulation. We can see as we let it run that the amount of virus in the simulation is tracking what the ODE predicts. The remaining exercise it's to add the cytokine field, and I'm gonna give people that have already finished some time to walk through, let's say 10 minutes, to walk through this exercise to try to get as much as possible in following this workflow. And the workflow is in the XML file, you already have as a placeholder when we imported the diffusion solver, you have the option of adding a second field. So what I want you guys to do is to adapt this code following the workflow that we did to add a cytokine field. Add IDs that would allow you to steer the diffusion coefficient and the decay constant of the cytokine from Python Steppables. Remove or comment out any other diffusion parameters or specifications from the field specification. Remove the secretion data, and then specify the periodic boundary. The boundary conditions are going to be periodic in the X and Y domain, and constant derivative in the C domain. And that would finish. That would be the last. That would allow you to specify the cytokine as a field in CompuCell. The next part would be to come to define the diffusion parameters in the Python Steppable. We define the diffusion parameters as a global parameter. In this case will be the cytokine diffusion coefficient. And then use the self get that XML element underscore element, the self that get underscore XML underscore element function to pass the data from the Steppable to CompuCell, from the Python script to the XML, both for the diffusion constant and for the decay constant. And just remember, as I showed, these have to be defined after you load the model and after you specify the scaling factors, because we're using those scaling factors. And lastly, we have ourselves secrete the cytokine, but in this case, the secretion is not, it's gonna happen if we look at in the model, secretion happens, which cells, this, if we look at the model, the cells that secrete virus, the secrete cytokine, are both the early infected and the virus releasing. So we need to iterate, in this case, through the virus releasing, and instead of just the virus releasing, we have to iterate over the over both the virus releasing and the infected cells. And then we have to load the cytokine secretor. We need to specify the amount that is going to be secreted, and that is specify the parameter P_C from the ODE model. And then we can actually have them, we can we can actually have them secrete the amount that they need based on the parameter values. Just, and as I said, I'm gonna give people 10 minutes to work on that. If people are experiencing problems at any step so far, please do ask to be put in a breakout room where people can work with you, on a one-on-one basis to be able to fix any problems that you might be encountering so far. So I'm going to, yeah, let's give until 2:10. So 2:10 Eastern, so people can walk through this exercise. Again, if people just want to start from here, people who haven't been working on the code, you can do that. You can do that as well by accessing the module _ 2.2 version 2, and then start from the virus code. Okay, so we're gonna create a poll here in a second, asking people whether they prefer to get a little bit more time to work on this, or keep going and then coming back later, because we have a version of the code uh, that um, so we're gonna right, because you can save the simulation as is right now, and then we can start over from the next part, uh, from the next version, and then you can come back by yourself later on to continue working on this exercise. Um, so and in the current poll, um, please answer done if you want to move on. Answer need more time if you want more time to continue to finish this one working, to continue working on this one. Okay, it seems like the majority of people needs wants to keep going. So I'm for those who want to work on this later, I'm gonna explain how to do it. You wanna save the simulate. You wanna save what you've done so far, and this is actually what I'm gonna do, because I'm not I did not work on it on this version. Um, just make sure that you remember what is the version that you're working on. You can close the project, um, go to CC3D, open project, and then we're gonna go into the folder that contains the module underscore 2.2 version 2. And inside that you have three options. Virus is the version that takes you up to to where we created the virus. Cytokine is a version that takes you up to the version where we added the cytokine. So this is the one that we're gonna start up from here. So everyone who wants to continue working with us in building the code, please do use this version, the module underscore 2.2 version 2, the cytokine version. Oh, I'm sorry, I forgot. I thought I was sharing my whole screen. Do you guys see my whole screen now? I'm sorry about that. Good. Right. So in the NanoHub directory, we are on module underscore 2.2, that version 2. And again, virus is the version where we added the virus. Cytokine is the version where we added the cytokine. And then we're gonna work we're gonna go from cytokine to loop now. So we're gonna start from cytokine here. And then people who wants to work on their version later, they can come back to the version that we're working on and finish this exercise. But to continue the module, we're gonna start from this one. If you were successful in if you implemented your version and it's working, you are more than welcome to continue working with your working version. So right, so the way we solve this again is in the XML file we specify. I'm gonna walk through it. I'm not gonna code it. I'm gonna walk through it really quick. We specify the cytokine field We specify the file, the field name. We added these IDs in the global diffusion constant and the decay constant, so we can steer them from our Python script. We specify the periodic conditions along the X and Y dimensions and the constant derivative plane value 0 for in the C dimension, the no flux and boundary conditions. That's all that we needed to do in the XML file. Next, we needed to add the specifications of the diffusion parameters. we added the cytokine diffusion parameter, the diffusion constant, and after specifying the and inside the start function, we use the we use this function that get underscore XML underscore element to pass on both the diffusion constant and the decay constant of the cytokine. The particular values of that cytokine diffusion constant are, again, arbitrary at this point, but those are also measurable experimental quantities. Next, we implemented the secretion by both infected and virus-releasing cells. And the reason we implemented it, we wanted both our infected and virus-releasing cells to secrete cytokine, is because we incorporated into our model the assumption that cells start signaling the immune system as soon as they get infected, even if they are not releasing virus yet. So the cell is infected, it has a virus inside, it's still not releasing virus, but it's already signaling the immune system that infection has occurred. We determined the rate at which they secrete this signal, the cytokine signal, from the model. It was PC. And then we then we loaded our cytokine secretor using the default snippet. We modified so it's a cytokine secretor, and the field name reference the cytokine. Then we determine how much the cell is going to release at each particular step, and then we divided that amount over the whole cellular domain. So the cell releases, and I'm sorry, there's a typo here. There should be cell, comma, and then the amount to be released at each pixel or at each voxel. That is going to be this amount. And the next part was plotting the integral of the field. We need to add another data series. In this case, it's going to be the met cytolocal. We call it met cytolocal because we have another cytokine signal, which we're interpreting as APCs. So we call this one local. And then we added it to our plot window that contains the total diffusive species, so we can overlay it with the ODE version. And then after secreting the cytokine, we counted how much cytokine we have in the system using the secretor that total field integral function that adds how much cytokine I have over my whole domain. But since we have 2 dimensions and the ODE is basically representing just 1, we have to divide by the number of dimensions that we have. We add those plots. We add that data we add those data, and then if we come back to player and we launch that version, which again is gonna be the one that contains the cytokine, if we launch that version, as soon as I get one cell infected, I should start seeing some cytokine being released. And now, instead of having two, I can actually visualize both the virus and the cytokine if I open a new graphical window, and in the pull-down menu, I choose the cytokine, and I'm gonna have another one to show the virus. The difference between these plots are definitely gonna be explained by the differences in their diffusion parameters, and in the fact that that we have cells secreting each of those. It's taking quite a little bit in my computer to run. Maybe might be running faster on yours. But at least I want to get to the point where we see some secretion of the cytokine. So see, there was a transition. As soon as that transition occurred, the cytokines started being released to the environment. So there might be, I do not see the cytokine being plotted. There might be a small error in that code. I'll promise that I will. It's not important at the moment, but I will check that version of the code and upload new. It's not plotting the diffusive virus either. Not exactly sure what's happening. I will come back to it and check it later. So this is an important question. Why are we looking at x1, xy1, and xy0 when we look at the diffusive fields? If I look on the level underneath, it looks a little bit more like this. I could do both, right? But the idea is that it is because of the way we are interpreting our model, right? You can you can see that we are interpreting the model as secreting the cells are secreting virus and cytokine to the extracellular domain, and that extracellular domain is the space that we created on top of the cells, that second layer. And that's why we're looking at layer 1 instead of 0, because we're assuming that they are being released into the extracellular domain that corresponds to layer 1. Okay. So okay, this next section so the idea is that we have already implemented the secretion of C and the secretion of virus. But now we want to connect all this together, having our cells, the transition between uninfected to infected, be determined by the local concentration of virus rather by the ODEs. So this is a refresher of what we did last time when we determined the transition from uninfected to infected. We slaved the cellular model to the ODE by reading of the virus V from the ODE model here. I am not gonna code this version. You can I'll show you how to load it, which is already coded, and then you guys can walk through this part by yourself by coding it as a homework exercise. But now what we're gonna do is we are gonna break this dependency of the cellular model on the OD model by having our cells get infected not by the amount of virus in the OD system, but the amount of virus locally that the cell gets locally exposed to. So instead of having B here, we will have a variable that describes the amount of virus that the cell gets exposed to. And the way we do that is we have this convenient function associated with the secretor called amount seen by cell. And that function tells you how much of the virus the cell is exposed to. So what we're gonna do is that instead of having the transitioning happening because of the virus, we're gonna have it happen because of the amount that the cell gets exposed to, which we're calling here seen amount. Okay, so we're changing we're breaking the dependency of the model on the ODE. Okay. Some of the parameters in that case need to be rescaled. If we do that, we need to rescale beta by some scaling factors. We're not going to discuss now why is that the case, but when we do that, when we break that dependency, we do need to rescale some of the parameters to ensure that we are reproducing the right the ODEs. So the version of the code when we implemented it looks just like this, right? But if we were to run if we were just to implement those changes and run, what will happen is that we will not get nothing will happen because we don't have any virus to infect any virus in the field to infect cells with at the beginning, right? Because when we start this simulation from 0 at our first simulation step, we have no virus, right? And since there's no virus, there is no cells that will get infected, so no cells will get infected later on, right? So the way the ODE model deals with that is that the ODE model has an initial number of uninfected cells, of infected cells. If you guys look at let's look at it here. If we look at the anti-money model, at the anti-money string, the anti-money string specifies that at the beginning of the simulation, we have P0 or 10 million infected cells, uninfected cells, and 75 infected cells. That means that in the original ODE model, 0.00075% of the cells are infected, right? We do have a problem there because if we were to start with the same number of infected cells in our model that we have 1,600 infected cells, we would need 0.12 cells infected to get the same number. But we cannot, the minimum number of infected cells that we can have in CompuCell is 1. So, because they're discrete objects, so we need to change the initial conditions in CC3D and the ODE model so we can then compare the both two models. The way we do that is that we determine an initial fraction of infected cells. And what this is gonna do is that once we have initialized our cells, we're gonna randomly choose 5% of our cells and we are gonna infect them. The way we do that is we randomly iterate through our cells. We pick a 5% of them and we infect them. In this particular case, when we start with 1,600 cells, we are gonna infect the 5% of them. It's gonna be 80. So we're gonna start with 80 infected cells. Now once we have decided that, we can actually update the ODE model at the beginning of the simulation so that we change the actual number in the ODE model as well. So we also gonna start with 5% of the cells infected in the ODE model, and in that case, that is gonna be 500,000. And then when we do that, then we can actually start seeing what the effect of breaking that dependency on the model is, on the ODE model is. And since I didn't code it, we're not gonna be able to work with that. But we do have a version that includes these broken dependency. The way we access that version is we go to CC3D project, open project, and then in module_version 2, we have 3 options: cytokine, virus, and loop. Virus is the version that implements the virus. Cytokine is the version that has both virus and cytokine. And loop is this version that allows you to break the dependency of the model on the ODE, the cellular model on the ODE. When we open that version, we see that it contains what I just walked you through. It contains an initial number of infected cells. It also contains the part where we infect our cells and the part where we change the initial conditions, so the ODE model, so they match the initial conditions on the CC3D model. And then we change also the infection rule such that here, I'm gonna explain this part of the code, 237 to 253. The original version we had was this one, where the infection rate is determined by the amount of virus on the ODE model. We have now implemented a version where the infection happens due to the local amount of virus, which is this version. And I talked about how certain parameters need to be rescaled, but the most important part is that this function returns how much virus the cell is locally exposed to, okay? And then what we're gonna do is, and we included this flag, use local virus. So if we set that one to, let's say no or 0, none. If we set that one to 0 or false, then we are back to the original simulation where we have the cytokine and the virus, but the infection rate is determined from the ODEs. Open simulation, loop. So if I run this version, the infection rate is determined from the infection rate is determined from the, we can see that there is virus already. The infection rate is determined from the ODEs. They look a little bit different because we have changed the initial conditions. And then if I set that one to true, the infection is gonna happen not because of the amount of virus in the ODEs, but because the amount of virus in the cell gets locally exposed to. So if I stop, and now we are introducing, there was a question about it earlier, we are in fact introducing the local speciality of the model. Okay. The last thing I want you guys to play with is to change that diffusion coefficient of the virus. Let's multiply it by 0.01. So we're making the virus less diffusible. And what we can see in that particular simulation is when we run it, is that the infection happens locally instead of happening globally. Instead of each cell being equally probable of getting infected, now the cells are getting infected by their local concentration of virus. The only problem, if I tell my Windows, so because I'm not using, I'm gonna do this. I'm not using this is because this part is important, right? So now we can see that the virus is much more local rather than being equally distributed, and that the infection is also much more local. That is happening, cells, instead of every cell having the same probability of getting infected, cells are getting infected by neighborhoods, if you want. The effect introducing the speciality has the effect of literally flattening the curve. We have talked about, it's been talked a lot about flattening the curve. So introducing this special component into the model has the effect of flattening the infection curve, because instead of cells getting, let's say, the virus of one cell being in contact with every other cell, the virus is just, cells are just infecting cells in their close, in their vicinity, right? So infection is gonna happen much, much slower. And this is the case where we can use this simulation to represent what happens in plaque assays, but also unintentionally, we can represent what happens when we enforce some types of containment measures like lockdowns or social distancing that that prevent the number of infections that happens due to, say, let's say, one cell of one person. And I'm gonna finish here, but I do want everyone to be able to run this simulation. Again, to launch it, you access it from it's in the version underscore 2.2 version loop. And this is the one that we're gonna start working from in the next modules. And if there's any questions, feel free to drop them in the chat now, and I'm gonna return back to James so he can talk a little bit about what's gonna happen next.